Good morning, everyone. I'd like to welcome you to today's webinar on the path to commercial success, 5 nanotech case studies, where Steve Oldenburg will share some of the nanocomposite story and examples of our customer success stories, which started just from an idea to a marketable product. I am really excited to hear the nanotech stories today, and I'd like to thank you again for joining us. My name is Rebecca Hart, and I'm the Regional Account Manager in the Sales and Marketing Department here at Nanocomposites, and I will be moderating today's webinar. For those of you that are not familiar with Nanocomposites, we're a 12-year-old privately held company located in San Diego, California, with product distribution worldwide. We focus on providing a wide range of precisely engineered and highly characterized nanoparticles and materials. We have one of the largest silver libraries in the world, and over 400 peer-reviewed publications have used our nanomaterial. Custom formulated versions of our standard products are also possible, and we work very closely with our customers and our partners to provide them with solutions to their projects and using our nanomaterials and our experience. So I'd like to now introduce Dr. Steven Oldenburg, founder and CEO of Nanocomposites. Steve?
Great, Rebecca. Thanks so much for the introduction. Yes, so today I'll be presenting a number of case histories where we talk about some of the internal projects and some of the companies that we've been collaborating with to bring nanotechnology-enabled products to market. And that really is our mission at Nanocomposites. So while we make materials, really the focus is in leveraging those materials to find some benefit of nanotechnology through the use of these precisely engineered and the highly characterized materials. The field of nanotech is not new. I've started my graduate work at Rice over 20 years ago, and back there was considerable expectations of the technology, and it's taken a lot longer than people thought to see those ideas result in real products that are being in the market and making an impact. But it is happening. So if we remember back in 2006, there was a lot of hype over the nanotech space, and there was over 1,200 companies that were started that had a nanotechnology focus, and not very many of them are still around. That said, there's some real and exciting applications on the horizon, and our main goal at Nanocomposites is to help accelerate those projects towards commercialization. So today's talk is the path to commercial success, 5 nanotech case studies. So I'll be giving a kind of a brief overview of the materials set that we use, and then some of the specific companies that we've interacted, that we've helped bring technology out to market. So in a lot of cases, it's not what you would have thought. It's not the sort of the standard things that you hear when you think of nanotechnology, but nonetheless, these are niche markets, but important markets, and more importantly, that these products can get out and be successful, and we're really excited to see where they go next. So in terms of the company, Rebecca talked a little bit about it, but we were founded in 2004. We've got about just over 40 employees, and with a really wide range of backgrounds, PhDs in chemistry, physics, biochem, and engineering. We've had over $20 million in R&D funding for the development of nanomaterials and nanocomposites, and the SBIR program, which is a government-funded program to help small businesses, and we've received a number of those in their commercialization index. We're in the top 10% for successful commercialization. We've got over 500 products developed. We're distributing through Sigma Aldrich, VWR, Thermo Fisher. We've got 2 groups in Japan and Europe that are helping us get products out there, and 15 other distributors. Currently working with 14 startups and corporations to help finish their R&D and bring specific products to market, and then we've got thousands of other customers that we've helped over the years in terms of providing materials or helping customize, doing small custom projects for them. We've got a number of patents that are issued and some pending, and we've licensed some patents in. So this is really our world. This is sort of the material space that we focus on. We've got metals and metal oxide core particles, primarily the center wheel shows the products or the materials that we use. Gold and silver are plasmonic, have really unique optical properties at the nanoscale. Iron oxide is a magnetic material that we use to give magnetic properties to the particles. Titanium dioxide is a high index photocatalytic material that can provide protection against sunscreens and also provide self-cleaning surfaces. Silica is an important scaffolding material for us, and then we've got copper oxide, which in conjunction with silver provide antimicrobials. Different surfaces can be put onto the particles to give a certain function, either to integrate them into a particular composite or to, in some cases, provide biofunctionalization and targeting. In addition to the core particles, we can also look at making a discrete shell that'll go around the particles. We do that with silica or other metals, and we can put those same surfaces on the surface of those particles. And then we've got this whole range of different sizes and shapes that we do with each of those sizes and shapes having a particular function or utility. So while we make materials, really our goal as a company is to help people leverage those materials into real commercial applications. So what happens is that we have this library. Researchers will purchase small quantities of the particles that we have in that library, and we don't have a lot of proprietary coatings. We provide a lot of information so that they can do real science. And when they're experimenting there, they often come up with something that works. And when it works, they usually come back to us, and they're needing either more particles or something a little bit different. And when we do that, when we come back, then we can really start to engage with them and to ask them a little bit more about what they're doing. And in that concept, when they come back, we can get down into what that idea is and whether we think it's going to be successful. We've had a lot of examples over the years where we've tried and in some cases failed to bring a nanomaterial over the bump and get it to a place where it's going to be commercially viable. And so when we hear a good idea, then we know it. And so the process of sort of making that decision, we have a criteria, and so our commercialization criteria is really, is there a unique nanotechnology enabled solution that provides, and one of two things, with the first being that you really need to have an order of magnitude improvement in the performance of an existing technology. If you're doing a 20% or 30% increment, that's really not going to cut it. By the time you get it to market, the existing technology will have caught up or surpassed you, and you won't really be making an impact. The second thing is that in some cases we're actually creating a completely new product category that was not possible without the nanotechnology component, and really that's the area that's most exciting. These are new applications that are only possible because of the unique properties, and the downside being that in some cases there's either not a market or that the market is relatively small because this is a new idea. So it does take a little while to bring that from concept to commercialization. The second point, which is sounds obvious but is actually really important, is can it be manufactured at a commercially viable cost? The materials that we're making are in some cases difficult to produce, often made in very dilute concentrations, and so when we actually look at putting them into into a product, we have to make sure that the value that they're adding is going to be sufficient to justify that price. But if these two components can be met, then we become really interested. And one of the things that's a little bit different about us is that when it sort of passes those two criteria, we then try to engage with the client in a different level. In some cases, that's dedicating an entire team to the application. Sometimes we'll help out with intellectual property and really kind of complete that IP strategy for the company. And in some cases, actually, in recently, we've been doing this quite a bit, is that we will actually become an investor in the company, and we'll take take an equity stake, and then really what that means is that we've got great alignment. The win is breaking things out to commercial, not necessarily just continuing the R&D, and that's worked out really, really well for us. So in terms of application areas that sort of meet those two criteria that I specified before, the first is biodiagnostics. We've got a bunch of lateral flow tests, imaging, sensing. Nanomedicine is another important area for us, topical therapeutics, drug delivery, and then sort of manufacturing under the controls that are necessary to get to nanomedicine. And then coatings, so optical and special effects colors. We've got flexible electronics, antimicrobial applications, and all three of those are examples where there is some value to the nanotech, and that in most cases we can meet the price point. So the first case study that I wanted to talk to you about is lateral flow assays. You would be familiar with these as sort of the over-the-counter pregnancy tests. And the way that they work is that you'll introduce some sample, whether that's saliva or urine or blood, put it in a little spot on this cartridge, and that will wick basically along the strip. You can see a picture of the strip down here. When it gets the conjugate pad, it releases some nanoparticles. The nanoparticles flow along the nitrocellulose membranes, and depending on what's in your sample, in terms of the analyte concentration that's in your sample, you'll either have a test line appear that indicates if the sample is present, or you can have a control line. Typically you'll always have the control line that just shows that it's working. And so what you might not have known from the pregnancy test is that those red lines that you're seeing are 40 nanometer gold particles. Typically they have that ruby red color in solution, and there's an antibody on the surface that's used to identify and attract the analyte and to measure a signal. And these are ubiquitous. Every drugstore has these on their shelf, and they work very well. Cheap, low power. They have their temperature stable, and very accurate and reproducible. So the question for us was, well, what can we add to this assay format? And one of the things that's really critical is sensitivity. So we had the idea of basically replacing the nanomaterials with the gold nanoparticles with a different nanomaterial. Instead of a solid gold particle, this would be a gold nanoshell. So these are made a little bit differently. You start with a silica core, and you stud a large amount of small gold particles to the surface, and then you use some chemistry that grows up those particles larger until you complete them into a complete shell. And the advantage of this is the particles are larger, but because they have that core that's glass, it's a little bit less dense, still retains the good flow characteristics, and results in a higher sensitivity assay. And that's important because in a lot of cases what happens is people are developing these assays, they work with the standard materials, and they just can't quite get to the correct analytical range. And so the test just basically doesn't work, and they can't commercialize it. They have to start over with something else. So this gives us that extra window of capability where we can really see very, very low concentrations of particles in solution and measured analyte. And one of the other things that we're interested in doing is not only just taking these strips and reading them by eye, but thinking how we might be able to quantify the output. And so usually that one of those lines, the test line, the strength of that line is directly correlated to the concentration of the analyte in solution. And in some cases, you don't want just a yes/no answer. You'd really like to know exactly how much analyte is there. And for that, you need a reader. And in the past, there's been these desktop readers that you can buy, but they're fairly expensive, and they're put in in one location. But we've been really thinking about how to integrate and use this sort of capabilities in everybody's cell phone to perform that same function. So we've been working with a number of different companies that have some versions that provide their own light, some that are passive, and then some that don't really even have a reader at all and just kind of rely on sort of the check reader type applications that people have on their phones in order to quantify that line. So once you have those two things, once you have this higher sensitivity and this and this lower this low concentration ability, we can go and do a number of different things. And so the first one application that we've been working on is for a newborn screening application. So when you've got a newborn, there's typically a blood test where you have a little prick and you get some blood and you test for a number of things. And one of the things that you can test for is it's called TSH, so it's thyroid stimulating hormone, and it's critical that those TSH levels are within a certain range. And if they're not, you can give a very inexpensive drug that'll bring it into the right range. But if it's not treated, it can result in mental retardation. And thousands of kids in third world countries such as India aren't diagnosed and end up with this disability, very easily corrected. And this test that we've been developing, coupled with a cell phone, would be able to sort of diagnose that at the point of care setting. Another application is stress. So we've been working with some companies that are looking to do salivary cortisol assays, and so that you basically would spit onto a strip and you would run the test and it would give you your stress level. And different people have different cycles of the salivary cortisol, and so what you need to do is establish a baseline and then look at differences from the baseline, but that can all be handled by the phone and by the app. And so applications here is just sort of making sure that people in high performance activities are at the top of their game, whether that's a surgeon or some military teams that are going out on a mission. Certainly interesting from an athletic performance perspective, and then also from the recovery aspect, where you're supposed to be taking it easy and relaxed. You can kind of monitor to make sure that that's the case. Another application that we've been working on in-house is in epidemiology. So we've been developing a tick assay for Lyme disease. So what you do is, if you've been bit by a tick in the Northeast, there's a huge problem. This is sort of the density of ticks that are found in the part of the States. It's really epidemic. Lyme disease is kind of a disaster if you get it, and very hard to cure if it's not treated. So what this allows you to do is basically take that tick, remove that tick, you would crush it up and put it into a small container, and then you would take that solution and run it on your strip. Put it in your cell phone. It would give you a reading and tell you whether the tick was infected or not. If it's infected, then you should get to the doctor, probably be prescribed antibiotics, and further sort of follow on that treatment. If it's negative, you're probably okay and don't need to pursue that. So the other thing that's interesting about the cell phone is when you do that test, it's easy to send that data up to the cloud. You can get sort of instant real-time mapping of infected areas. So the data becomes in some cases just as valuable as the test itself. Another test we've been working on, this is one of the SBIR grants that I alluded to before, and this one is really just looking for whether you've been exposed to something. So we developed a multi-organ injury assay. So basically it looks to see if anything is, if your heart or your liver or your kidney has been damaged by something. And this was really the problem in the Gulf War, is people were out in the field, they came back, they were sick, they don't know what they were exposed to, but it was something that's caused them some harm. So the idea here is we can't test for thousands of different chemicals, but we can look at your body's response from a particular from an organ perspective, and just see if you've been hit with anything. If you've been hit with anything, then you can immediately be removed from that situation and be triaged and into some sort of a treatment. So that was sort of on the diagnostic side. A second case study is on medical dermatology and anesthetics. So this is in conjunction with a company called Sienna Biopharmaceuticals. Here we're using the thermal properties of nanomaterials. So we've got with basically a silver plate that has basically it acts as a resonator at a particular frequency. That's a function of its size and its shape. So we can tune that geometry for these particles to strongly absorb in the infrared, so either out in the sort of 800 or 1000 nanometer regime. We coat it with silica, which provides a way to stabilize it in the body or in a high concentration solution. And what we do is, what the process is you'll make this high concentration solution, and then you'll basically rub it on the skin. And there's really two areas, initial applications. One is for the treatment of acne, and the second is for light hair, laser hair treatments. So for the acne treatment, what happens is this very dense solution of these nanoplates gets rubbed on the skin. There's a sort of a vibrating device that massages those particles, and they'll take those particles inside that hair follicle and get it close to where the sebaceous gland is. And when you get particles close to the sebaceous gland, you hit them with that near-infrared light. The particles will heat, and they'll damage some of those sebaceous cells. And what that does is that basically modulates down the oil production. And once you've kind of permanently turned down oil production in that acne lesion, that acne lesion will clear. The laser hair treatment is very similar, and then here you're sort of damaging the heat, or using the heat to damage the stem cells at the base of the hair, and that can sort of turn off hair production. And that's something that's useful, because typically when you're going in for laser hair treatment, you need that dark pigment in order to absorb the light. So if you don't have dark hair, they basically turn you away and say that it's not going to work. And so here's a way that you can kind of bring that in and make that happen. So exciting for a number of reasons. I believe this will be one of the first plasmonic therapies that's out in the commercial market, looking to hit the market sometime next year. And you know, that really provides a stepping stone from the FDA's perspective of establishing safety, and then looking to do sort of more complicated you know, nanomedicine type applications, where you might have an active biopharmaceutical, or even have a sort of a drug delivery injected type of application. The third case study is a company called Viveware. This is a Dr. Marcus Duda, who's out in Florida, and had, you know, many years in wound care and foot care as an orthopedic surgeon. And what he was frustrated with was sort of the standard options available from bandages, and had this idea that he could do something that would be better. And so the premise is that when you've got these wounds, these venous stasis ulcers that are wounds that basically won't heal, and some people have these open wounds for years and just can't get rid of it. What he developed was a treatment that would basically provide advantages over the standard of care, which is sort of wrapping it, or a foam bandage where you can pull off some of the exudate, but then it ends up sort of wicking and filling that bandage. And then he found a way to really provide some sort of amazing developments in terms of healing. So this was 11 weeks post-treatment, and what the device was a sock, and compression socks are actually used already in wound care. And typically what happens is that wound will be bandaged up, and then you'll put the compression sock over top of it, and that'll increase circulation and sort of help with the healing. What he did was different, is that he basically proposed using a compression sock, but without the bandage, and allowed the sock itself to be the bandage. So the socks are made out of wool, and what they do is they will actually wick away that exudate from the wound, and allow the wound bed to remain moist but not be saturated. And that sort of controlling over that relative humidity or the moisture content right at that wound bed actually makes a huge difference in the healing rate. One of the problems is that since it's open to the environment, there's the potential for drop-in of microbes or bacteria that could cause infection. And so what we worked with Marcus on was adding a silver nanoplate to these socks. So the nanoplates have a flat side. They adhere strongly to the wool fibers. And because of their very high curvature at the edges of the particles, they continuously release silver ions. So they provide this for long-term protection against antimicrobials. They also sort of can have a positive impact on the wound environment itself, and sort of mitigate and be complementary to the increased blood flow of the pressure and the moisture monitoring or the moisture control at that wound surface. A fourth case study, these are a magnetic tracing particle that we did in conjunction with a company out in Texas. And here what the problem was is that they were basically trying to understand what was going on in a extended fracking well environment. And so here you've got tens or even hundreds of these well sites. You're injecting fluids down into the earth, and it's really important that you understand sort of the underground flow of water or of the different reactants that are in that fracking well. And so what happens is that you want to be able to trace it. You want to be able to take a sample of water at one location and know whether, hey, did this come from this well or if it come from this well? And so there's tracers that are out there. But they typically only have sort of 5 or 10 flavors. And what these guys wanted is something incredibly sensitive that would have a very high degree of multiplexing. So what we worked on was a new formulation of magnetic nanoparticles that are encapsulated with a very robust shell. And so these dark centers here are magnetic nanoparticles. What we do is we build the nanoparticle, we change the charge of the nanoparticle, and then we add DNA and basically make a DNA layer on the particle, and then we encapsulate that DNA layer in this aluminum silicate shell. And that aluminum silicate, sort of like a glass, it's almost like the dinosaur amber preservation that people find, where they can go back after hundreds of millions of years and extract some biological component because it's got this protective shell on it. So this survives harsh environmental conditions. Sometimes the temperatures are over 100 degrees C at the bottom of in these wells. And then what's amazing is, due to the ability of us to use PCR to amplify that DNA, basically a teaspoon of this material is sufficient to detect particles in 100,000 gallons of water. So we can have just a very small amount introduced at each well stage, and then we can sample days later and say, oh, we've got 40 different detection particles that we can measure, and we've got this ratio, and because of this ratio, we kind of know this is where the water's flowing. So what if we increase the pressure over here? And then if we increase the pressure over here, that has the potential of getting out another 10,000 or 100,000 gallon barrels of oil out of the ground. And so that was the concept of that particular case study. The last case study, again, completely different. It's a company we've been working with that's really been thinking about one-way illumination. And what we mean by one-way illumination is this is a thin coating that basically gets applied to a window. And when you look at that window from one side, you see a bright patterned illumination, basically almost like a neon sign that's glowing at night. But when you look at it from the other way, you don't see anything. And so this is the concept here, is that you could produce these thin films, you could put them on windows, on buildings, they could provide advertising from the outside, but it wouldn't block the view or cause any issues with people on the inside. There's a second application will actually be our first product, which is, instead of doing a whole window, we're just going to make glasses, and these glasses will have that same coating on the front and will basically be illuminating a pattern to the outside, but when you look through it, it looks normal. And the other thing about these glasses is that they have a battery in them, so we'll have a lithium-ion battery and Bluetooth connection, and it can all be connected to the DJ. And so there's these sort of self-illumination clubs in Asia where you can have everybody wearing these glasses at the beat of the music or the control of the DJ, sort of have this pulsing phenomena. So sounds a little bit of a novelty, but there's a lot of people that have been really interested and excited about different variants of that technology. And the nano-enabled component here is that the way these films are made is basically you have a nano imprint, so a very high density imprinting of spots into a thin film. And then we developed a particle that has a surface that will self-assemble into those wells. And those particles have a high index of refraction. And so when you edge illuminate, send light in or inject light from the sides of the thin film, you can get light out in both directions. You get little scattering centers, and if we metallize or otherwise coat one side of it, we can create this one-way illumination configuration. So again, sort of, need these particles to be small so that they self-assemble, and it provides a potentially very cost-effective way of making these sheets and assays. So that was really what I wanted to do, is just give you a broad overview of some of the things that we're working on. Again, not what you typically think of as nanotechnology applications, but there really is a reason why that technology is important, and we've been able to find ways to make these in a cost-effective format. One of the things I just wanted to finish up with was that there's a real need when you move to commercialization to make these particles be reproducible. So we have over the last few years, we've really focused on our quality system and building an environment that's sort of appropriate for making these materials, especially as we move into the the therapeutics and sort of drug delivery nanomedicine space. All right, so with that, if you let me know if you have any questions associated with the talk, and I'd be happy to address that.
Thank you, Steve, and thank you to our audience. As Steve mentioned, we're going to go ahead and take some of the questions that people have submitted during the presentation. If you haven't already, I'd like to encourage you to use the question pane on the control panel, so we can address your question during our live presentation. The first question today comes from Brian. Could you briefly explain how the environment can affect the plasmonic property of the nanoparticles?
Sure. Yeah. So there's a couple of aspects to that question, and I mean, one is what effect does the particle have in the environment? And we've done a lot of work in the nanotox space to ensure that these particles are safe and have sort of low impact for human health and our surroundings. In terms of stability, long-term stability, it depends on the material. So for the plasmonic materials, we typically use gold and silver. Gold is incredibly inert and generally has very robust properties towards the environment. The stained glass windows in some churches are gold nanoparticles, and they've lasted for thousands of years without degrading in color. Silver does oxidize and is affected by light and other characteristics. Sometimes that silica shell that we put on can protect that material, and that's one of the reasons why we use that in that acne treatment to protect against acid and other etching environments that can potentially be out there. And in a lot of cases, when we're looking for very robust particles, we've actually recently developed methods of gold shelling silver particles, so very thin, even just a couple of atomic layers of gold on that silver particle. It doesn't affect the plasmonics that much, but gives us that added stability. That's something that's relatively new, but if you're interested, please contact us, because we can do that on most of the silver particles that we have.
Great, thank you, Steve. We're going to go ahead and take another question from Paresh. A lot of the focus is on nanotechnology. It's obviously on the size of the nanomaterials. What is the role of the zeta potential of the nanoparticles for their biomedical application?
Sure. So zeta potential, which is basically the surface charge of the particle, is extremely important, especially in biomedical applications, and that's because as soon as you introduce a particle into a biological system, that particle is going to undergo some type of a transformation. There's going to be something in solution that's other things in solution, whether those are proteins or cells or other chemical components, that's going to interact with that surface. And typically what people talk about is forming a protein corona. So it basically has a whole nother layer that's on the particle that may be completely different from when the particle started. And that protein layer is really what dictates the fate and transport of that particle. So controlling and understanding that surface in a biological system is critical. And the zeta potential, so how that particle starts, and charge is a very important component of that, will influence that final protein corona, or that final terminal layer that's going to dictate its performance in vivo or in vitro systems.
Great, thank you. Another question today from Alan. Are there any FDA issues related to medical use of your nanoparticles?
Right. So it's a really interesting question, because for a long time people thought that nano might be different, and that we might have to change how we're studying these materials, change how we're approving these materials, because that small size could be doing something surprising with biology that we hadn't anticipated before. So there was a whole field of nanotoxicology that got started to basically address that question, and more than $1 billion has been spent sort of investigating whether these particles are safe and how they interact with biological systems. And the short answer is that there really hasn't been anything different that they found. There's nothing really surprising about engineered nanomaterials that have led to a surprising toxic result. So the rules for approval for these materials are basically the same as they are for any other chemical or compound that's already being studied by the FDA. So you have an idea, you've shown some efficacy in some nanomedicine application. The first thing you need to do is make sure that you can reproducibly manufacture that particular thing, and show that you're going to be able to do that over and over again. And then you take that substance and then you run it through the standard testing that's done. So pharmacokinetics, safety reactions, and then you go through that phase 1, phase 2, phase 3 trial phase to make sure that it is both safe and efficacious.
Great, thank you, Steve. I also have another question from Ray. If you could briefly outline the steps for developing a lateral flow assay, and then to kind of coincide with that, asking about the detection limits, if the detection limits can reach picomolar sensitivity for proteins.
Right. Yeah. So the lateral flow strips are simple conceptually, but there's a bunch of details that have to be controlled in order for them to be successful. So this is essentially what it's made of. So you start with a backing card that just holds everything together. You have a little sample pad, and that's where the sample gets applied to. And then this conjugate pad is where the nanoparticles are. And so you'll have typically the nanoparticles. When you say conjugate, that means that you've got an antibody particle combination, which is referred to as a conjugate. And that is, you make those conjugates up and then you dry them down on that conjugate pad with a sugar, so that when this liquid hits that conjugate pad, those particles are released. And when they're released, those particles start to flow down this nitrocellulose strip. And on this test line here, there's another antibody that's striped onto that test line. And when the analyte is present, so you've got your particle with the analyte, when the analyte is present and captured by the particle, this antibody will recognize another piece of that analyte, and those particles will then bind to that test line. And when they bind to the test line, you'll start to see a color form due to the color of the nanoparticles themselves. They'll continue up, for the particles that don't bind, they'll continue up the hit control line, and that just captures the remaining nanoparticles to make sure that the assay is working. And this wicking pad at the end is just designed to pull everything across. So that's sort of the basic concept of what we're doing. And certainly picomolar concentrations are possible. For a lot of compounds, we're getting down to picogram quantities, and there's some variants that'll go even lower than that. But there's always this sort of limit of sensitivity. There's other instruments that will be more sensitive, but they just won't be as cheap and as fast and as quick and as convenient as lateral flow. So the further we can push that sensitivity limit, the more utility these types of diagnostics will have.
Great, appreciate the thoughtful answer on that, Steve. Do you currently have any materials that are being used in an FDA-approved product? If so, if you could explain what those are.
Yeah, so we've worked with a number of lateral flow companies that have our particles in commercial assays, so they're certainly being commercialized there. The Sienna Labs application that I talked about earlier with the acne and the light hair treatment, that's the closest thing that we have to commercialization. So they're just finishing up their phase 2 pivotal trials, and they'll be able to uh, get the data for the 510K that'll allow them to commercialize that next year.
Perfect, thank you. This is from ProCrush. Is the scale-up of nanomaterials similar to the scale-up of small molecules or biologics?
Yeah, so I think different in most cases. Typically what we do is most of the materials that we make are in water, and that's just because from a waste stream perspective and sort of environmental concerns, waters are a really great solvent for us. They do get made at relatively dilute concentrations, so what happens is that we end up making relatively large volumes, either hundreds or in some cases thousands of liters of these materials, and then we have to concentrate and wash them. So we have these sort of tangential filtration loops that will perform that function, and then we can purify and concentrate them down to a place where they're going to be useful as a sort of a master batch that can be integrated into composites or be used for different types of biomedical applications after we've put some functional surface onto the particle.
Great, thanks, Steve. Another question comes from Biho. Are your materials patented, protected, and/or proprietary?
Yeah, so a lot of our base materials are not, in the sense that we basically we have recipes for making them, and then we can get them out to people so that they can use them without restriction. And I think that's, for a lot of people, that's really important because they don't want to get tied down to something specific, and they just want to kind of move forward with the science. But we do have a number of pieces of IP that provide us protection on some of the aspects and some of the applications of the different materials. So using the nanoshells in lateral flow, we've got some IP around that aluminum silicate shell that I alluded to in the magnetic application. One of the things that we found is that silica, just the regular standard Stober silica, has a relatively high dissolution rate, so it'll dissolve in certain environments and disappear. And that aluminum silicate really turns it into a clay and makes it much more robust. And so we've got some IP around the things that are kind of critical, and especially when we're thinking about productizing or bringing something to market, we try to find at least something that's going to give us that protection if we go to market, so we don't just get copied.
Okay, that's all the questions I have. So I'd like to just thank you again to our audience for joining us. It's really a pleasure to have you and submitting the questions and intently listening. If you have any further questions that we didn't address today, you may reach us at either the email provided below in this slide or the phone number. We'd be happy to address any questions that you have. And then really just like to thank you again for joining us, and we would like you to have a great rest of your day. Thanks so much.